Good evening, students. Time is going. So everybody's the maximum members are explaining about how to study, and they are giving so many strategies for you. So kindly select any one strategy. Which one is suitable for you? Follow that strategy alone to prepare to crack a Neet exams. So don't confuse yourself, because that strategies are created based on the own skill. So kindly select a suitable strategy. So anyhow, you have to prepare well. You have to prepare well to crack the Neet exam. Clear? So study well, revise well, and repeatedly attend that MCQs yourself. Clear? So yesterday we discussed about the immunity, types of immunity, acute and innate immunity and acute immunity. So in last topic we discussed about the lymphatic organs. So lymphatic organs are matured in the lymphatic organs. Lymphocytes are matured in the lymphatic organs. The two types of lymphatic organs are present. One is a primary lymphatic organs, another is a secondary lymphatic organs. So that up to that level, I explained. The next one is a primary lymphatic organs. Primary lymphatic organs. So primary lymphatic organs are major sites of lymphopoiesis. Here, lymphoid stem cells are proliferated, differentiated, and mature into the immunocompetent cells in the absence of antigenic stimulations. The primary lymphatic organs are production centers and army headquarters of the immune system. The mature lymphocytes of primary lymphatic organs migrate through the blood or lymph, which is accumulated into the secondary lymphatic organs. Here, those cells are exposed to the antigenic stimulates and becomes a functional by production of a specific antigen antibodies, where in secondary lymphatic organs. Clear? So up to this level we discussed. Today I have to explain the primary lymphatic organs with the suitable examples. Clear? So first one is so the 3 is a best example for the primary lymphatic organs. So what are they means? The first one is a thymus in the case of birds, bursa of Fabricis, and bone marrow in mammals. So thymus and bone marrow in the mammals. Clear? So the two things are important on thymus. So thymus is a primary lymphatic organs. Thymus is a training center for the army of T lymphocytes. So thymus, the first letter is a T. Thymus, first letter is a T. The thymus responsible for the production of a T lymphocytes. Clear? So the two cells are responsible for the immunity. One is the T lymphocytes, another one is a B lymphocyte, shortly called as the T cells and P cells. Thymus, first letter is a T. So which is responsible for production of a T cells. Similarly, B cells, bone marrow cells. B, first letter is a B. So which is responsible for producing the B cells. Clear? Like that. In this way, you can easy to remember it. That question may be asked in Neet, thymus T cells. That is a code word. So that thymus is a army center of T lymphocytes. This you can see the structure of thymus, ma. This one, this is a thymus. The thymus resemblance a time of leaf and leaf. Hence, which is named as a thymus, which develops from the epithelium, which is developed from the epithelium. Thymus begins its development in the 6th week of gestation. At the time of birth, it weighs around 15 to 20 grams. The time of birth, the thymus weight is only the 15 to 20 grams and increases its size after birth. It increases that after birth. It reaches about 40 gram weight by puberty. Thereafter, it will slowly atrophies. So initially that weight is low. So after birth, it increases its size. It reaches up to 40 grams. In human beings, thymus consists of two oval lobules. Two oval lobules. It is having the two lobes, which is oval in shape. In birds, several lobes are present. In human beings, only the two lobes are present. The thymus is covered by the fibrous, which is called capsule. It is formed of two lobules. The thymus covered by the capsule. The capsule is formed of two lobes. Each lobe of thymus is organized into lobes which are separated from the each another by septae. The septae is a separating layer which is called tuberculae. Septae is called tuberculae. Within the lobules, the cells are arranged into a outer cortex and inner medulla. That cortex is tightly packed within the proliferating immature lymphocytes. The cortex consists of the lymphocytes. Lymphocytes means thymocytes, because thymus are present in the lymphocytes present in the thymus. That's why that lymphocytes are called thymocytes. That cortex consists of the thymocytes and reticular cells. The lymphocytes are multiply rapidly from the thymic lymphocytes. I mean T lymphocytes, which causes the cell-mediated immunity. So important point in thymus means thymus is a primary lymphoid organs. That is the most important question. Thymus is a primary lymphoid organs. That is a most important question. Here T cells will be developed. That is a second important question. T cells developed from the thymus. That is a second important question. The third one is the T cells are responsible for the cell-mediated immunity. Clear? Cell-mediated immunity. The three things are important questions from the thymus. Clear? So this level is enough for you, because the thymus, each one is a big chapter. The thymus alone we can discuss up to 2 hours. Clear? For Neet point of view, this level is enough. The next one is the functions of major functions. The T cell maturation, cell-mediated immunity, graft rejection. Graft means you know very well, while that organs will be transplanted. T cell maturation, you know very well, the T lymphocytes, I mean the responsible for immunity, that's well, and that cell will be produced and matured in the thymus. The second one is the T cells are responsible for the cell-mediated immunity. The third one is the T cells is responsible for the graft rejection, means organ rejection. So, for example, kidney transplantation. So, at that time, the rejection surely will be occurs, because the own antigen, own cells will be accepted by the human bodies. Clear? So, when one kidney is failure means, at that time, what we do? Automatically we are transplanting the another kidney. We can get it a kidney from the any donor, from family members, otherwise from outside. So at that time that kidney is considered as to be transplanted. Kidney is considered as foreign things. So at that time rejection may be occurs. That the T cells are responsible for the graft rejection. Clear? So we will discuss in the transplantation side, the graft rejection. So graft rejection means organ transplantation time rejection will be occurs. At that time suppressor, etc. At that time we can take a treatment. After that we can do the transplantation to avoid the rejection. Clear? So those are the important things of thymus. The next one is bursa of Fabrysis. So bursa of Fabrysis is a primary lymphoid organ in birds. That is the important thing. It is not in mammals. Bursa of Fabris is present in the birds, which is also the training center for the army of B lymphocytes. So the bursa can able to produce the B lymphocytes in birds. The B lymphocytes develop for stem cells. The B cells in birds, keep it in your mind, bursa of Fabris will be present in the birds. The B cells develop from the stem cells present in the egg yolk, liver, bone marrow. So that bone marrow, egg yolk, and liver. So from that organs only, liver or bone marrow, the B cells will be developed. B cells will be developed, and which is also matured. Which is also matured in the bursa. Bursa means a sac-like lymphoepithelial structure, which is attached to the dorsal side of colicia. Bursa are responsible for mediating the humoral immunity. That maturation will be takes place in the bursa of birds. Clear? Up to this level, Anafma. That important question is a bursa of Fabris is a primary lymphatic organ in birds. That B cell maturation and humoral immunity will be takes place in the bursa of Fabris. That is important questions. The third one is bone marrow. Bone marrow. So bone marrow is a primary lymphatic organ, which is a soft tissue located within the cavities of. This is a center part. You can see the arrow mark. So a bone marrow is a primary lymphatic organ. It is a soft tissue which is present in the located in the cavities of bones. I mean center part of bones. So you know very well, while if you are taking a non-veg means, at that time you can take a bone marrow. So at the bone marrow, center part of that bone marrow, center part of the bone marrow having the very soft tissues. Sorry, that soft tissues of bones having the bone marrow. That bone marrow is a production center for the lymphocytes. Protection center for the lymphocytes for B lymphocytes. That bone marrow responsible for the production of a B lymphocytes. Approximately 3 kg in an average human adult. Bone marrow is a key component of a lymphopoietic system. Bone marrow is a key component of a lymphatic system. It can producing the lymphocytes that will be supported the body's immune system. That will be supported by the body immune system. That bone marrows are three types of bone marrow tissues. What are they? Means a red bone marrow, yellow bone marrow, third one is a stromata. So red bone marrow means red bone marrow, which is consist of a hematopoietic tissues. Here RBC, WBC plate cells are arise from the red marrow. So that is important question. Kindly note it. WBC, RBC and platelets arise from the red marrow. So I will explain only the important thing. Already told this is a big chapter only. So in Neet point of view only, I will give the points. So red marrow responsible for producing the RBC, WBC and platelets. RBC, WBC and platelets arise from the red marrow. Clear? In birth, all bone marrows are red. when increasing the age, at that time the red marrow converted into the yellow marrow. The second one is a yellow marrow. So yellow marrow is called medulla osseum flava, which is mainly made up of fat cells. Yellow marrow is made up of a fat cell. That yellow marrow consists of the blood vessels and capillaries. Yellow marrow consists of blood vessels and capillaries. That is important thing. The third one is I am giving only important points. Stroma. Stroma is a tissue of bone marrow. Stroma is tissue of bone marrow that is not directly involved in hematopoiesis. Hematopoiesis means bone marrow production. So directly involved in the hematopoiesis. Yellow marrow makes up the majority of stroma. Yellow bone marrow make up the majority of stroma. Only few or some red marrow cells also consist of the stroma. Clear, ma? So bone marrow means what are the important Neet question from bone marrow means bone marrow is a primary lymphoid organ. This is the first one. The second one is the bone marrow is the production center for the B lymphocytes. That is a second important question. That bone marrows having the three types of bone marrow tissues. One is a red marrow, yellow marrow, and the third one is a stroma. Red marrow means that RBC, WBC, and platelets are arise from the red marrow. Yellow marrow means the blood vessels. The yellow marrow consists of the blood vessels. Stroma means tissue of bone marrow. Stroma is a tissue of bone marrow. That stroma does not directly involved in the hematopoiesis process. Clear? Those are the important points from the bone marrow. The next one is the secondary lymphoid organs. So secondary lymphoid organs means concerned with the immune reactions. Concerned with the immune reactions. Already I told that primary lymphoid organs are responsible for the production of some lymphocytes like T lymphocytes and B lymphocytes. T lymphocytes will be produced from the thymus and B lymphocytes will be produced from the bone marrow in human beings. Clear? So that lymphoid cells will be produced and it becomes a functional one in the secondary lymphoid organs. So primary and secondary both will be supporting for the immune systems. Clear? So secondary lymphoid organs are concerned with the immune reactions. Immune reactions because that is a functional one. The secondary lymphoid organs are lymphocytes are made functional. So made functional, just now I told secondary lymphoid organs, the lymphocytes, I mean T and B lymphocytes are made functionals. They are army campus of immune system. The secondary lymphoid organs of army campus of immune system. The cells are exposed in the secondary lymphoid organs. The cells are exposed to the antigens and they produce antibodies and sensitized cells. Here only in secondary lymphoid organs only the antibodies will be produced. The lymphoid organs are small and poorly developed. The secondary lymphoid organs also small and poorly developed at birth. They grow progressively with age. When that age is increasing, at that time simultaneously that secondary lymphoid organs also growing. So what are the examples means? Examples means the most important secondary lymphoid organs are lymph nodes, spleen, MALT. Lymph node, spleen, and MALT. So we will discuss one by one. The first one is lymph nodes. The first one is lymph nodes. So lymph nodes are a secondary lymphoid organ. This is a structure of lymph nodes. Nodes means a small knot-like structure. This is called as a node. Node. We can say node. Lymph node. So like a small knot-like structures. So lymph nodes are a secondary lymphoid organs. They are complex cellular spherical or a ovoid ovule structures present along with the lymphatic ducts. That human lymph nodes are a bean shaped. That is important Neet question ma. That human lymphoid organs, I mean lymph nodes. The human lymph nodes are a bean shaped. Approximately 1.25 mm in diameter. Human lymph nodes are bean shaped. It is about 1.25 mm in diameter. One side of lymph node has a intention, the hilus, where the blood vessels enter and leave the node. Several afferent lymphatic ducts carry the lymph. Here. This one. Several afferent lymphatic ducts carry lymph into the gland, and single efferent lymphatic ducts, different lymphatic duct leaves the node. The lymph nodes act as a filter and filter the lymph. It is an important question ma. The lymph nodes act as a filters. The function, the mostly it will be act as a filters, and they filter the lymph. That each lymph nodes are made up of three regions. What are they? Means the cortex that is present in the outer one. Outermost structure is called as a cortex. This one. And middle paracortex. Middle is a paracortex, and inner is a medulla. And inner is this one. Inner is called as a medulla. The cortex consists of the B cells. That outermost one. That cortex consists of the B cell. Cortex consists of the B cells, and the paracortex contains T cells, and the medulla contains both B cells and D cells. Important question ma. In secondary lymphoid organs, in lymph nodes, cortex contains B cells, paracortex contains T cells, and the medulla inner part consists of the both B cells and the T cells. So inner part only having the two cells B and D. Outer only having the B. Clear? Those cells group together and form a nodes or a primary follicles. I think this level is enough, ma. I already told this is a very depth topic only. Up to this level, enough for you. Because if I go depth means surely you will confuse. You will make a confuse. Up to this level only enough. The second one is the spleen. Second one is the spleen. So spleen is also secondary lymphoid organ. It is a solid encapsulated organ located in the upper part of the abdominal cavity. Upper part of the abdominal cavity, behind the stomach and close to the diaphragm. I think already you studied in 12th. The spleen is a secondary lymphoid organ. You know very well. It is a solid one. That spleen is a encapsulated organ which locates in upper part of the abdominal cavity, behind the stomach and close to the diaphragm. The spleen is a component of lymphatic system. The spleen is a component of a lymphatic system. Kindly take a notes, ma. So after completion of this class, you can revise it easily. You can understand that points. Clear? So spleen consists of a efferent lymphatic vessels. Efferent lymphatic vessels. It is a deep red in color. Spleens are a deep red in color. That spleen has direct communication with the main arterial circulation. The spleen has direct communication with the main arterial circulation. It contrasts to the lymph nodes, which filters the lymph. The spleen also filters the blood. The spleen also filters the blood. This question may ask, which one filters the blood means which lymphatic organs filters the blood? That is a question means your answer is the spleen filters the blood. The spleen surrounded by the capsules. The spleen surrounded by the capsules. That capsules penetrated into the tissues as a septa, which is called trapeculae. The capsids penetrate into the tissues as a septa. That septa is known as a trapeculae. There are two distinct regions in the spleen, namely as outer red bulb and white bulb. Outer red bulb and inner white bulb. So red bulb is outer, white bulb is inner. That red bulb consists of the large number of blood-filled. That red bulb consists of the many number of blood-filled sinusoids in which phagocytosis and plasma cells are found. Where? In red bulb. The red bulb is concerned with the destruction of old and dead red cells, dead red blood cells. The white bulb consists of the lymphatic tissues. The white cell consists of the lymphatic tissues. The cellular arrangements of the white bulb was first described by Malpighi. Hence, which is also known as a Malpighian follicles. That is important question mark. That white bulb of spleen is also known as a Malpighian follicle. That white bulb of spleen is also known as a Malpighian follicle. The major part of lymphatic tissue is arranged around the center arterial is known as a perianterior lymphatic sheath, abbreviated as caps letter PALS. That PALS consists of a B lymphocytes. B lymphocytes grouped in small masses called follicles. So follicles means many things are grouped in a particular place. That is called as a follicles. Here also PALS consists of the B lymphocytes grouped in a small masses called follicles. In between the follicles, distributed the T cells, the T lymphocytes. The T lymphocytes are distributed in the B lymphocytes, I mean the follicles. The B lymphocytes follicles may also consist of the germinal center. Germinal center, when it is known as the secondary follicles. I think this level is enough for you. So what are the major function of the spleen means? Formation of RBC at the time of necessity. At the time of necessity. So when that the lack the RBC production from the major site, at that time that RBC will be produced from the spleen, and spleen another major important function is filters the blood. One more thing, the spleens can bring the both humoral immunity and cell mediated immunity, because that T cells is present in one region, outer region, and B cells are present in the inner regions. T cells are present in the inner region. That both B and T will be present in the middle region. So it can brings the both humoral and cell-mediated immunity. Those are the important points, ma. Next one is the immune cells. So immune cells only. Just now you know the what are the cells comes under the immune cells. That level is enough. Because this is, it is not present in your NCERT book, but this is related to the immunity. So immunity means we should know that antigen, antibody interactions, immunoglobulin, lymphatic organs, immune cells, etc., etc. But lightly you have to know about that one. Outline you should know. Because in 1 or 2 times they will ask questions from the outline of lymphatic organs and immune cells also. That's why I am explaining about the immune cells and lymphatic cells. At least you should know what is what. Immune cells. The cells concerned with the defense of the body are called immune cells. What are the cells concerned with the defense of the body? The collection of the cells are called immune cells. The immune cells are the army of defense system. Army of defense system. They are soldiers of the immune system. That immune cells are responsible for the immune system. That's why which is known as soldiers. That functions as a sentries and produces the weapons and toxins. So anyhow, it will be act as a fight with the antigens. So what are the important immune cells means lymphocytes. So naturally B lymphocytes and T lymphocytes are immune cells, and null cells, NULL, null cells, natural killer cells. That question is also previously asked. Immune cells, natural killer cells. I think that is also present in your syllabus. Now, NK cells, shortly called as NK cells, natural killer cells. And another one cells called K cells, means killer cells. Clear? Those are the comes under the lymphocytes. What are the lymphocytes are present? B lymphocytes, T lymphocytes, null cells, natural killer cells, and killer cells. That 5 cells are comes under the lymphocytes. And the second cell is the macrophages. Already I told macrophages responsible for the phagocytosis. We discussed yesterday class also. Clear? The third one is a dendritic cells, eosinophils, basophils, neutrophils, antigen presenting cells, master cells, platelets, mast cells. Those are the example of immune cells. So among the cells, lymphocytes, the 5 lymphocytes and macrophages, mast cells, platelets, antigen presenting cells. Those are the important immune cells. Kindly keep it in your memory. What are the important roles of immune cells? Means immune cells defend the body against the pathogens. Immune cells defend the body against the pathogens. The natural killer cells kills infected cells. And that question is the most important one, ma. The natural killer cells kills the virus infected cells and cancer cells. The natural killer cells kills the cancer cells and virus infected cells. So don't make a confusion, ma. That virus infected cells can able to produce the one cells which is called as the interferons. Clear? That is a different one. Here that natural killer cells kills the viral infected cells and cancer cells. That macrophages kills the pathogens by a phagocytosis. Next one, antigen presenting cells watch the body like a sentries. Antigen presenting cells deposit the peptide fragments of pathogens on their surface. Mostly that surface only that having that marker, etc., etc. Because that surface only that antigen antibody reactions will be takes place through the binding on. So that antigen presenting cells deposit the peptide fragments of the pathogen on the surface. The peptide fragments of pathogens are recognized as the T helper cells and TC cells, TH cells and TC cells. TH means T helper cells. Clear? The T helper cells role is activates the B cells. T helper cells. Why it is called as the TH helper cells means T cells which mediates the cell-mediated immunity, but H means a helper. Directly the TH cells not involving the cell-mediated immunity, it will be supports, it helps to the kill the antigens. However, the T helper cells activate the B cells. What we do? Automatically B cells will produce the antibodies. That antibodies kills the antigens. Clear? So that T helper cells directly not involving the neutralization of antigens. T helper cells activate the B cells. That B cells produce the plasma cells. Plasma cells produce the antibody. Antibody will neutralize the antigens. Clear? That is also important question mark. The B cells produce the humoral immune response by the production of antibodies. TC cells brings about a cell-mediated immunity. Those are the important things. Those are the important points and role. Those are the important role of immune cells. Next one is lymphocytes. Lymphocytes means just now I told the WBC circulating the blood and lymph. Mostly that 5 types of lymphocytes are B cells, T cells, and killer cells, natural killer cells. Clear? That lymphocytes brings the immune response. Lymphocytes bring the immune response. Remaining thing I already explained. T cells are responsible for the cell-mediated immunity. B cells are responsible for the humoral immunity. I mean, humoral immunity is also called as a antibody-mediated immunity. Antibody-mediated immunity. The next one is the B lymphocytes. B lymphocytes are a WBC maturing in the bone marrow in mammals. B lymphocytes are a WBC maturing in the bone marrow of mammals. That it will be circulated in the blood and lymph. B cells containing the surface immunoglobulin is called BCR. BCR means that cell surface having the some places, that place for binding site of antigens, that is called as BCR. BCR. B cells containing some marker like binding site, like that is called as a BCR. They brings about the humoral immune response. Similarly, the T lymphocytes mature in the thymus. It is also circulate in the blood and spleen. Here also is the T cells also containing the surface receptor called TCR. Receptors means that binding sites, like a binding sites. The particular thing will be like a lock and key. How that key will be, specific key is responsible for open the particular specific lock. Clear? All keys are not fit to the all locks. Clear? So one key here, one particular lock having the specific key only. Like that T cells also. Like that T cells also containing the specific receptor. That receptors are present in the surface of T cells for binding of antigens, which is called as a TCR. That brings the cell-mediated immunity. Which brings the cell-mediated immunity. Next, important functions you should know. T helper cells, just now I told T helper cells are a class of WBC. T helper cells are a class of WBC. will be supporting the stimulate the B cells, indirectly supporting the immune system. And that NK killer cells, that the function also I told that memory cells. Memory cells means it consists of the CD4 and CD8. That memory T cells are produced by the activated T cells. That major function of memory T cells means name itself it having the answer. Remember the antigens which attacked or activated them. Memory T cells are remember the antigens which activated them. For what purpose means the mount a quick action when the same antigen infects again. So what is the important function of that memory T cells means they mount a quick action when the same antigen infects again. Clear, that is the major role of memory cells. At least you should know what is what. Null cells. Null cells are lymphocytes which having the cytotoxic property. Null means a nothing. Cyto means a cell. Toxic means a toxin. Cytotoxic. Toxins, cyto. Cyto means a cell. Toxic means a toxins, unwanted things which may harmful to the particular cell. That is called as a toxic. So null cells are lymphocytes with cytotoxic property. They are neither B cells or a T cells. The null cells are intermediate between the T cells and B cells. The null cells are intermediate between the T cells and B cells. They form a third population cells. That's why which is known as a TPC, third population cells, shortly called as a TPC. The two types of null cells are available. One is a killer cells, another is a natural killer cells. This specific small chapter is present in your Neet syllabus. NK cells, natural killer cells. Natural killer cells are a group of null cells. They form a third population of lymphocytes, shortly called as a TPC. Those non-pathogenic lymphocytes having the large granules, hence the cells also called as a larger granular lymphocytes. The cells show a natural cytotoxicity and they can kill the range of tumor cells, means cancer cells, and cells infected with the virus without any antigen stimulation. So when the cells having the tumor and cells infected with the virus, clear. So that cells shows a cytotoxicity in naturally. That is the important point. They themselves identify the tumor cells and viral infected cells and kill them. Just now I told that NK cells, NK means a natural killer cells. It identifies themselves without any support. It is an independent identification. It is an independent identification without any support of other cells. It can able to identify the cancer cells and viral infected cells. After identification, what we do? It will be kill that one because if tumor cells grow, what will happen? It is dangerous to our health. No. So that's why the tumor cell should be a kill. Similarly, viral infected cells, if you are not control or we are not destroying that viral infected cells, means that virus will be multiplied and it will be infect the another cells near the infected cells and near the infected cells. So all cells will be virus infected. So all functions will be collapsed. That's why the tumor cells and viral infected cells will be destroyed by the natural killer cells through the cytotoxicity property. Clear? They do not need any help from the other cells. Hence, they are all named as a natural killer cells. Independent, due to the independent action of that one, which is called as a natural killer cells. That natural killer cell originated from the lymphoid pronuclear along with B cells and T cells. That natural killer cells are differentiated and matured into the bone marrow, lymph nodes, spleen, and thymus. After maturation, they enter and circulated throughout the body. The natural killer cells are components of both innate and adaptive immunity. That is also important, that natural killer cells are components of both innate and adaptive immunity. Those are the important points. What are the major function of natural killer cells? Means apoptosis. That is an important question, apoptosis, the killing of virus hidden inside the cells. Killing of virus hidden inside the cells. Hidden means hiding property. Clear? So killing of virus hidden inside the cells. Lysis of virus infected cells. If one cell is affected by the virus, means that cells will be lysis. Lysis means killed. Next one is lysis of tumor cells. So just now I told viral infected cells and tumor cells will be a lysis. Viral infected cells secrete cytokines. Cytokines activated the NK cells because cytokines are signaling molecules. The cytokines are signaling molecules that cytokines are activate the natural killer cells. For example, one cells are affected by the virus means that viral infected cells produce some signals named as many signals. So one of the signals is cytokines. That signals will be activated the natural killer cells. That activated natural killer cells will secrete the gamma interferons. So already I told viral infected cells will be produce the interferons. Viral infected cells will be produced the interferons. Here that gamma interferons, shortly called as IFN. IFN means gamma interferons produced from the viral infected cells. So that IFN activates the macrophages, which leads the phagocytosis. In this way, the lysis the viral infected cells. Can you follow me, ma? It is a very simplest one. Don't make a confusion, ma. The first one, just to imagine it, one particular cell is present. That virus will be entered. That entered virus will infect the particular cell. So after that, now the cells are infected with the virus. So that viral infected cells will produce the cytokines. The cytokines is a signal, act as a signal. So that signal activates the natural killer cells. That natural killer cells secretes the IFN, gamma interferons. That gamma interferons will activate the macrophages. That macrophages are which leads to phagocytosis. Inside that phagocytosis, what will happen? Lysis, because lysosomes are present in the phagocytic cells. So automatically the lysis of a virus. Lysis of a virus infected cells. Clear? This is an important one because just now I told NK cells are present in your Neet syllabus. The NK cells kill the body cells possessing a low level of class 1 MHC molecules. MHC means a master histocompatibility complex. Leave it that points, no issues. Because it will make confusion for you. Because I have to explain what is MHC1, functions are, etc. etc. That is a two depth. The NK cells also recognize the tumor cells, not only the viral infected cells, also recognize the tumor cells and kill the tumor cells also. So the activation of natural killer cells results in the extracellular release of certain chemical substances into space between the two cells. I mean infected and non-infected cells. The most important of such a substance are a perforin or a cytolysin. Kindly note it, ma. That perforin and cytolysins are chemical substances which are released by the natural killer cells. Clear? Perforin and cytolysin, which lyses the virus infected cells and causes the death of the virus. Clear? That is the important question. The next one is the killer cells. Killer cells also second type of null cells. They form the third population of lymphocells. Already I told they are antibody dependent. Killer cells are antibody dependent. In NK cells mostly dependent the T cell dependent. I mean cell mediated dependent, because the lysis process will be takes place. Once the lysis process, macrophages or phagocytosis, the three words are involved, means it is a cell mediated. Here the cells only lysis the particular thing. Clear? So that is a cell mediated one. But in the case of killer cells, K cells, antibody dependent one. Dependent one. So here that cells, that K cells possess a FC receptors for binding with the immunoglobulin G antibodies. Hence, they can bind with the cells coated with the IgG antibodies and automatically it kill them. Automatically it kill them. This level is enough. And macrophages, so many times I told, is a large molecules derived from the monocytes. That macrophages derived from the monocyte, and macrophages involved in the phagocytosis. That level is enough. I think next one is that's all about the immunity. So immunity chapters, you must know what's the meaning of immunity and the types of immunity: innate, acute. Acute means active and passive. Active and passive both are subdivided into the natural and artificial. Both active also having the active, natural and artificial. Passive acute immunity also having that natural passive immunity and artificial passive immunity. So after that, you must study about the lymphoid organs, primary lymphoid organs, secondary lymphoid organs with the examples. The next one is immune cells. Clear? So after that, you should know what's the meaning of antigen and antibody. I gave definition for antigen antibody. I gave definition for antigen antibody. Although you should know some other important points about the antigen and antibody. Then only we can understand the antigen antibody reactions. The first one is the antigens. First one is the antigens. So antigens are a substance which is introduced into our body, which stimulate the production of antibodies. The most antigens are proteins, but some are carbohydrate. The most antigens are proteins, but some are carbohydrates. Lipids are nucleic acid, and already I told antigens are foreign particles, which cause or stimulate the antibody production by our immune system. That antibodies mostly may be carbohydrate or maybe proteins or lipids or nucleic acids also. That antigens may be proteins, mostly proteins. Some of antigens are carbohydrates or lipids or maybe nucleic acids. That antigenic determination, which is called epitope, that is an important Neet question. Antigenic determinant is called as a antigenic determination, or epitopes are those sites of antigens that are recognized by the antibodies. Clear, ma? That antigenic determinants are called as epitopes, because that epitope and paratope, the two words are important. Ma? That questions will surely be asked in the Neet exam from the antigen and antibody topic. Antigenic determinant is, which is also known as epitope. What's the meaning of epitope? Means epitopes are sites on antigens which is present in the particular antigen that are recognized by the antibody. That epitopes will be recognized by the antibodies. And the receptor present in the T cells and B cells, which is known as epitopes, based upon the ability of antigens to carry out their functions. Antigens are two types. One is a complete antigens, another one is a incomplete antigens. the based on the ability of antigens to carry the functions. So based on the carrying of functions, the antigens are two types. One is a complete, another one is a incomplete antigens. Incomplete antigens. So complete antigens means able to introduce antibody formation and produce specific and observation reaction. So complete antigens are introduce antibody formation and produce specific reactions with the antibody, which is called as a complete antigens. Incomplete antigens means which is also known as a partial antigens. Incomplete antigens also called as a partial antigens, which is known as a haptens. That is an important question. Haptens, H-A-P-T-E-N-S. Haptens means the incomplete antigens. Which are haptens are a substance which are incapable of introducing antibody formation by themselves. So incapable of introducing antibody formation by themselves, because antigens means it should be stimulate the antibody production. Clear? So that the complete antigen only, without any other influence of other cells, it can able to stimulate the antibody production. But in the case of haptens, it cannot capable of introducing the antibody formation by themselves. Clear? That's why which is known as a haptens, partial antigens, but can be capable of inducing antibodies on combined with the large molecules. Large molecules means most probably proteins, which serves as a carriers. Clear? So that some antigens cannot stimulate the antibody production. Certain antigens will be combined with the proteins. Here that protein will be act as a carriers. That antigens will be carried by the proteins. After that, that antigens will be after that, that antigens will be stimulating the antibody production. So which that such antigens are called as haptens. So haptens are important questions, ma. So red blood corpuscles of ABO blood groups possess a common antigens, the H antigens, which is a precursor for the formation of A and B antigens. This example for red blood cells of ABC blood group possess a common antigen, the H antigen, which is a precursor. The H antigen is a precursor for the formation of A and B antigens. Because A blood group having the A antigen, B blood group having the B antigens for the production of opposite antibodies. B means A antibody, A blood group means B antibodies. Clear? So that's all about antigens. That's all about the antigens. That antigen presenting cells also important one, because that antigen is should be present to the any other things. That is called as antigen presenting cells. Clear? So what are the examples of antigens means? Viruses, bacteria, fungus, some toxins. Those are the few examples of the antigens. Proteins, lipids, nucleic acids. Those are the example of antigens. What are the antigens? Where the antigens are present means in ABO blood groups, antigens are present. We already discussed in Mendelian ingredients. And RH blood group antigens. RH blood group antigens. And MHC class 1 molecules antigens, MHC class 2 molecule antigens, HLA, human leukocyte antigens. Tumor antigens will be deposited on the tumor cells, and viral proteins will be deposited in the surface of a viral infection. The ability of material can introduce the immune response, which is called as the immunogenicity. Immunogenicity, that materials are known as immunogens. So what are the materials will introduce or will support the stimulation of immune response, which is called as immunogenicity? That materials are called as immunogens. Clear? The antigen having the epitope, just now I told, which is comes under the structure of antigens. That is the most important one. So antigen, epitope means nothing, surely easy to remember. Antigen means the antibody binding site. So antigen, epitope is present in the surface of antigen. Epitope is present in the surface of antigens. In that epitope, only that antibody will be binded. So that antigens, epitopes, antigenic determinant or epitopes means nothing. That antibody binding site, that is called as a epitope. That is important on ma. That antigens has many epitopes. Many epitopes. That many epitopes, I mean that antigens with many epitopes are called multivalent antigens. Many epitopes means many epitopes of antigens, which is called as a multivalent antigens. What are the chemical nature of antigens? Means it is a proteins or a nucleoproteins. So surely one question may be asked from the immunoglobulin or antibody or antigen sites. So again, I give the summary for the antigens because that is an important one. Because immunology means you should know about the antigen and the antibody thoroughly. Again, I repeating the substance it can induce the specific immune response is called as antigens. That it can react specifically with the antibody. That is the nature of that antigens. That antigens are a partial antigens called as a haptens. The here proteins will be supporting to carry the antigens. Incomplete, complete antigens means that antigens directly stimulating the antibody. That is called as the complete antigens. That the ability of antigens to produce the immune response and react with the products, which is called as the antigenicity. That the ability of antigen to produce the immune response and react to the products, which is known as the antigenicity. And what's the meaning of immunogenicity? Means the ability of material to introduce the immune response is called as a immunogenicity. The next one, the substance which induces the immune response, which is called the immunogen. The substance which induces the immune response is called as the immunogens. That what are the antigenic substances? Means viruses, bacteria, fungi, toxins, proteins, polysaccharides, lipids, nucleic acids, and so on. Binding site, which is called epitope, which is also known as the antigenic determinant. When the epitope is present in many, which is called as a multivalent antigens. Different types of epitopes are present in the antigens. From the single antigens, different types of antigens will be present it. The number of epitopes in an antigen is called valency of antigens. That is also important. What is the meaning of valency? Means how many number of antigen is present on the cell, which is called as a valency. Valency means the number of antigens. Number of not antigen, number of epitopes present in antigens. It is called as the valency of antigens. And heterophil antigens occurring in unrelated animals. Heterophil antigens are identical and they exist on the surface of cells, which is surface of cells of unrelated plants, animals, and bacteria. So that antigens are majorly 6 types. What are they? Means that based on that function only, that antigens are 2 types. One is a partial and complete antigens. Based on the presence of antigens, location of antigens will be classified into the 6 types. What are they? Means exogenous antigens. Exogenous means allo antigens and autoantigens. Both are comes under the exogenous antigens present in outside, autoantigens and allo antigens. Next one is endogenous antigens present inside, and thymus independent antigens. Another one is a thymus dependent antigens. Next one is happens. Last type is a cross-reactive antigens. Those are the 6 types of antigens. The immune cells scan the body for the antigens, pathogens. So without antigens, cannot develop the immune system. That antigens are recognized by the B cells and antigen presenting cells. That is an important question, ma. That antigens are recognized by the APC. APC means antigen presenting cells and B cells. B cells and antigen presenting cells recognize the antigens. That antigen presenting cells first recognize the antigens and submit to that antigen to the B cells and T cells. T helper cells. Again, I repeat, once that antigen is entered means that the antigen presenting cells will recognize that antigens. After recognition, it will that recognized antigens says submitted to the B cells and the T helper cells, because T helper cells also supporting for the B cell activation. After that, B cells are produced the antibodies by the plasma cells. Clear? So the B cells bring the humoral immune response and T cells bring the cell-mediated immunity. That thing already explained. What are the major function of antigens means immunological reactions. Immunological reactions, and that antigens only brings the humoral and cell-mediated immune response. That antigens brings the allograft rejection. Allograft rejection, it determines the blood groups. That antigens only determine the blood group, A blood group, B blood group, or AB blood group, or a O blood group. That ABO blood group is determined by the antigens. That thing also already we discussed in the code. Already we discussed in the multiple allele system and another one ingredients. Topics, ingredient chapters we already discussed. So it brings a graft rejection also, allograft, specifically allograft rejection, because that antigens are present normally. One foreign things is entered, for example, transplantation side, it will be rejected based on the antigens. So it brings the allograft rejection. Clear? Those are the importance of antigens. I think this level is enough, because antibody structure and functions, if I explained means I have to explain that immunoglobulin also, because yesterday also I told all immunoglobulins are antibodies. If I start that one means it will take a more time. So that immunoglobulin structure and function we will discuss on tomorrow. Today class is over. Thank you, ma. Thank you very much.